I did find an earring after class last week, like somewhere around in here on the floor. So if anybody, you know, it's one of those ones that doesn't have a back on it. So yeah, lost an earring. There's one up here. So good morning, and I wanted to share with you just the overall results of them on Wednesday. Not your usual bell curve, but we did have live days, and the mean was a 75, which is by far the best mean that I've ever had on any first exam. Of course, I know that there's some of you that didn't do as well as you hoped. I would encourage you to change something before the next exam so that you just don't have a repeat of that. Maybe, you know, come talk to me. Let's think about how you were studying, maybe what you can do different. Maybe you just need to double down and, oh, I didn't realize I had to study more. Maybe that's all it is, but don't do everything the same, expect to get a different result the next exam. So let's try to be proactive. Maybe you can try to get a tutor. I know sometimes those are in short supply. Come see me more often. By all means, come and look at your exam at some point in my office. I'm free after class today. If that's a good time for you to stop by. It's hard to get more than 2 people in my office at once, but sometimes we can make that work. And I don't usually have a big rush of people, so come see me, come over, look over your exam. I am going to share I think 4 questions with you next time. I only had about 5 people doing makeup, but I have 1 person yet to do that. So I'm not going to share those with you today, but I am going to share the most commonly missed ones. But it's always good to look that over. There were several of you that didn't bubble in your username. So you wrote it, but then the Scantron can't read your writing, it needs to read the bubbles. So bubble in your username. About 10 of you didn't bubble in your test form. The computer knows which one form you're more likely to have, but it just takes me a little longer. Also, write your name on the actual exam. And then when you come back and look at it, you can easily see, you know, which ones you actually put on it without having to go back to the Scantron. I also had several people that skipped a question on their exam. I was able to go back to their exam, see what they circled on there, and a few people got, you know, credit even though they totally skipped it. So, good idea to put your name on the exam as well. Okay. All right. So, unless there's any questions, we'll move on here. After the exam, and like I said, I promise I won't do that future exam. we started chapter 12, which is kind of it's not a very lot of material that I have to share with you about eukaryotes because it's not our main focus in this class. But we did talk a little bit about classification and protozoa, a little bit about malaria, and just really briefly about algae. So you kind of are going to want to know kind of the main differences between, bacteria and archaea and protozoa, algae, fungi. Those are all kind of the microbes that we're thinking about. And I do want to talk a little bit about fungi today. There's some terminology to learn. But first, let's think about significance and primary characteristics. I'm getting a little feedback. Is that bothering anybody? Because I'm recording, so okay, good. All right, so fungi, what's the main thing to know? So we're talking about, of course, mushrooms are fungi, molds, and yeast. All right, obviously mushrooms are not microscopic, so that's not our main focus here. I did change my picture here just to a picture that I had taken myself last fall. Obviously, there's lots of mushrooms that are fungi. Last fall, there was a week where the conditions must have been just right. It was cool and very wet. And like walking through the woods, there was this huge fungi that just totally burst through the forest. So that was kind of cool. So we are mostly interested here in molds and yeast. Just a second. This is bugging me. Let me see if I can maybe get into this one. Okay. All right. So what are the primary characteristics when we think about what makes a fungi a fungi? Well, thinking about the structure, cell walls. There's some variation there, but very often the major constituent of cell walls is chitin. So that's a polysaccharide, kind of like cellulose is in lots of green plants. Chitin is formed in fungi. And probably the main thing that will stick with you is that fungi are not photosynthetic. Okay, they are never, that's just kind of a difference between plants and fungi. Okay. Significance. So why would we be interested in fungi? Why would we want to study it? Give me some examples of why we might be interested in fungi. Well, for one thing, we eat it, right? Some people do. Mushrooms, not for me. But a lot of people like mushrooms. So we certainly eat mushrooms and other truffles, that sort of thing. What else? Yeast. What do we use yeast for? Yes, baking. Okay. Again, the food. Also, fermentations. Yeast fermentations, right? Alcohol. Right. We think about mold as being something detrimental, right? It spoils our food. And we don't like that, but some molds produce very important things for us that has helped us fight infection. Okay, there are some molds that produce antibiotics. Okay, so drug production, that's pretty important. There are also very important decomposers. Okay, so meaning they are recycling nutrients. They are kind of using dead things as nutrients and breaking them down in the environment. Okay, that's why you mostly are going to find mushrooms in the forest on dead or dying trees or other organic material. Let's see, what else? Certainly there. Oh yeah, you got one. So is it just molds that produce antibiotics, or do mushrooms and yeast produce antibiotics? I believe just molds. I don't know of any mushrooms or yeast that do that. Okay. We will talk more about that in 3 or 4 weeks from now. What else? Certainly, there are some fungi that cause disease. Things like athlete's foot, something that maybe is not, terribly severe. Yeast infections. There are certainly fungal diseases of plants too. And other very more serious, lung infections. Fungal diseases are harder to treat because they're eukaryotic cells and they're more similar to our cells. So, could be a problem. And last thing I wanted to mention is that fungi can have symbiotic relationships with plants that are often overlooked and actually quite important. We think most plants have these symbiotic relationships. The term for that is mycorrhizae. Mycorrhizae. Okay, so that's probably a new term. These are fungi that grow in association with the roots of plants. Okay, so that's this little graphic up at the top here. I just got that, I think, from some kind of lawn company advertisement. But we don't realize this happens, but we know that most plants have symbiotic relationships with fungi, with their roots. The fungi, again, we call this relationship mycorrhizae, grow in association with the roots. They help the roots absorb water. And in return, those green plants provide some nutrients to the fungi. So apparently, if you grow grass without those mycorrhizae fungi, it's not growing as robustly as when the grass has those symbiotic relationships. Kind of hard to tell the difference between the grass and that picture, but we think this is very common that a lot of plants have this. All right, so how fungi helps us to some extent, maybe in some cases how it hurts us. So we want to talk through some terminology that we use to describe fungi. And again, we're talking both about filamentous fungi, that's the mold type, as well as the yeast type. So we're going to talk first about basically the molds. And those filaments, we have a more technical term for those, we call those hyphae. And we actually, filaments of cells. Okay, so we actually have several different ways that we would describe that hyphae. They can either be septate or aseptate. Okay, septate means that there are cross walls between the cells. And aseptate, that A means without, so without cross walls. So in this graphic that your book gives here, it's kind of hard to tell the difference here, just looking at it. But the one on the left here, you can kind of see these cross walls and the filaments here. Okay, so like here's one cell, here's one cell. And then the one on the right here, the aseptate hyphae, don't have those cross walls. So essentially, each hyphae is like one big multinucleate cell. Okay, so when something is multinucleate, many nuclei, we have another term for that, senescent. Yeah. Okay, so there's another term, senocytic. That's a cell that has many different nuclei. So, nutrients and everything just kind of flow there. With the septate, there's actually little pores in those cross walls so that we can share nutrients and gas exchange and things in between cells. Okay, so hyphae is going to be one or the other, has cross walls or doesn't have cross walls. And then they're also going to be considered either vegetative or aerial. Okay, so vegetative hyphae, their main function is to obtain nutrients. And when you get big masses of these vegetative hyphae, then we call them mycelium. Okay, so large masses of hyphae, or of vegetative hyphae. Right. And then aerial hyphae, these are your reproductive structures. And they are going to give rise to aerial spores. In this latest edition of your book, they pretty much cut back all the material that talked about reproduction in fungi. So that was fine with me to drop all that off. But just to mention, in terms of how do fungi reproduce, all fungi will do asexual reproduction. So basically, these spores that come off of these aerial hyphae will germinate and grow into a new filament. They all produce these spores asexually. Some fungi also will have sexual reproduction where the spores that are made are not exactly the same as the parent. Okay, so there's two different organisms. There's recombination, and then the parent or the daughter filaments are not exactly the same as the parent. Okay, but they all do sexual reproduction again, and then some kinds will do asexual. Sorry, they all do asexual, some will do sexual. And we used to still do to some extent classify fungi based on their reproductive structures. Okay. So I know some terms in there, I'll review them in a minute, but here's an example of a fungal mycelium. Right? So masses of these hyphae, often they're intertwined. The old version called this a scanning electron micrograph. The latest version has fixed that. If you look at your units here, that's something you can see with the naked eye. This is a great big mold, so it's actually supposed to be a light micrograph there. And then I just pulled this off the internet because I just introduced all these new terms, and this is just showing some of them a little different way. Here's a mycelium, right? A mass of vegetative hyphae. Senocytic hyphae. There's hyphae that don't have the cross walls. Septate hyphae. Here's an example of an aerial spore structure. And so the reproductive structure grows off of the vegetative one, and then when the time is right, those spores will ripen and be released. Sometimes, when the wind blows or whatever, and then those spores will germinate. The hyphae will grow out of that spore. I'm going to talk about this sporium in a minute, so we'll get to that. This is just showing, here's these aerial spores on top of a vegetative hyphae here. And this is showing one of the most, usually the way yeast reproduce, and that's by budding. So we're gonna actually talk about that, some yeast structures in just a second here. All right, so that was the filamentous. Moving on to the yeast, they are small. We could describe them as globular. Every once in a while, we see these when we're looking at something else in the lab, and yeast are going to show up as a little bit bigger than bacterial cells, and they're not perfectly round. Because when they reproduce, they usually reproduce by budding. And so that just means you get kind of an unequal, when a bacterial cell divides, it usually divides right in half and you get two equal daughter cells. But with budding, just a small portion of it kind of pinches off from that first cell. If that bud doesn't detach all the way, then we would call that a pseudohyphae. And this was the picture of the old version of your book. The new version shows a different picture and doesn't emphasize the structure as much. But that's showing, it's kind of hard to see this. So this one right here, the bud is still kind of attached to that. So we think that that might be a mechanism that yeast can kind of fit into, in between cells, maybe to some extent, when it kind of just has this smaller bud sticking out. Okay, so helps penetrate between cracks or between cells. And just know that some yeast are going to be, or some fungi are going to be able to go back and forth between that yeast-like morphology and a more filamentous morphology. So we call that dimorphic growth. Okay, so let's say the organism exhibits filamentous and yeast-like growth. And typically, which morphology it takes on is going to be dependent on the environment that it's in. So it goes by a number of environmental clues, maybe temperature is one of them, maybe some other things like oxygen concentration, things like that. But typically, it's not very often going to be doing both of them at the same time. So in this picture, you see both the kind of roundish yeast cells as well as the filaments. Again, it depends which environment it's in. One fungi, Candida, is a good example of one that can do both. So Candida causes infections like thrush in the mouth, athlete's foot, a lot of different things, and it's quite flexible and takes on a lot of different forms. And it's not as easy as you would think, like, oh, in this situation it's always doing this, and in this situation it's always doing that. So where do fungi get their nutrition? This is a term that we're going to pick apart in a minute when we just start chapter 6 in just a minute. But for now, know that most of them are chemoheterotrophs, and that just means that they are going to get their nutritional requirements from things that they ingest. And those are going to be organic sources. So we will pick that term apart in a little bit as to what those several prefixes mean. Many fungi are considered saprophytic. And that just means that they absorb their nutrients through things that are dead. Okay, so that's the easiest way to absorb nutrients because you know, like a log or whatever that's dead, the cell walls have started to break down. It's very easy to penetrate through another organism or a plant that's dead. Okay, so saprobes do that. Some are going to be considered parasitic. And that just means they get their nutrients from living organisms. So at the expense of some other living organism. And those that do that are usually have a modified, are able to make modified hyphae called Hostoria. Okay, so this is the best picture I could find of that over here. Basically, this fungal hyphae, again, it's just going to be a little bit more beefy hyphae that's able to penetrate into living cells. Okay, so it's just modified a little bit to be a little bit tougher. Right. And really, the last thing that I have to say about fungi here, when we have a fungal disease, we usually refer to that as a mycosis. You see that prefix myco, and that usually often means we're talking about something to do with fungi. Okay, so like I said, when we have mycoses, they're just harder to treat because they're eukaryotic cells, and it's harder to get some drug that's going to be only targeting the fungi and not hurting our cells. Okay. So that's pretty, like I said, I'm not going to get into the reproduction anymore. There's a number of terms there that I want you to be familiar with. Any questions, comments about fungi? Yeah. Okay. Oops. All right, so let's move on to chapter 6. So we already dipped into chapter 6 a little bit. We already we had a short assignment about culturing bacteria and media a little bit in the last exam. We're going to go back and pick up the rest of that chapter where we're thinking specifically about what kind of things microbes need to grow. So specific elements, for instance. We'll spend just a brief amount of time on that. And then think more about, if we want bacteria to grow, what kind of environment do we need to provide? What kind of temperature? What other things play into the growth of microbes, like oxygen concentration and pH and things like that? Thinking about counting cells, can we figure out how many cells are going to be in a culture maybe 3 hours from now? Yes. So we're going to walk through that a little bit. We're going to talk about growth curves, what growth typically looks like, and then some different methods of actually counting cells. And so if you're in lab, you have done one of those methods already, the viable plate count. So hopefully we will get to that before you have your lab exam, either today or Wednesday. All right, nutrients. So macronutrients, thinking about the elements that bacteria cells and basically any kind of cell would need to grow. We call them macronutrients. So that just means nutrients that are needed in large quantities. Okay, macromolecules, those are things like proteins, carbohydrates, lipids, and nucleic acids, right? These macronutrients then would be used to make a cell's macromolecules. So some of the obvious ones here that cells need in large amounts: carbon, hydrogen, oxygen. We're just going to say cell structures because they are in just about everything. You know, cell walls, cell membranes, proteins, all that. And along with nitrogen, things like nucleic acids, right? Nitrogenous bases, adenine, guanine, they're very rich in nitrogen. Amino acids. things that have a lot of nitrogen. Okay, so probably 95% of the cell is made up of those 4 elements. Okay, so cells need a lot of those. Also things like phosphate, nucleic acids. Obviously, we have a sugar, a base, and bonded by a phosphate in there. Phospholipids, so cell membranes. ATP has phosphate, so phosphate's very necessary. And then these other elements, potassium, magnesium, calcium, these are also important cofactors for enzymes. So that just means we need a lot of those in order for enzymes to function. And I have iron here in parentheses because sometimes iron is considered a macronutrient, sometimes it's considered a micronutrient. Iron is found in hemoglobin in our cells as well. So again, there's just some disagreement whether to say that we need a lot of it or a little of it. It's somewhere in between, I guess. But cells also need micronutrients. Sometimes those are referred to as trace elements. No less important for cells. They just need them in very, very small quantities. Okay, so we'll say needed in small quantities. How small? Usually, like what's in tap water would be enough. Say zinc or manganese or chromium. That would be enough for a microbiological medium. Okay, so just very small amounts. Again, these are needed as cofactors in order that enzymes can function properly. And then there's a lot of variability as to what kind of organic things or growth factor a cell would need. Remember, something's considered organic if it has carbon to hydrogen bonds. Okay, so something like carbon dioxide is not considered organic. Bacteria often can make many of the amino acids they need themselves. We as humans can manufacture 10 of the 20 amino acids. Bacteria usually can do a little better than that. They can make the ringed ones, the aromatic amino acids as well. E. coli, for example, can make all 20 amino acids, but there is some variability there. And I should say they can make make this organic structure out of inorganic elements. But sometimes, you know, cells need some coenzyme like NADH or some kind of vitamins in order to support growth, but a lot of variability. All right, so that brings us to this figure that, coming back to these terms that we use to describe how organisms get their nutrition. All right, so these terms pull together several different prefixes and then, you know, put them in different arrangements. So it's sort of inherently complicated. I'm not a big fan of these terms, but it's one of those things that you just kind of got to be familiar with. We can describe how an organism gets its energy as well as where it gets its carbon. Okay, carbon is just kind of like one of those big things that obviously it needs a lot of. And we put these prefixes together along with the root troph. So this term troph means nutritional, or let's say nutritional. Or sometimes it's, I wrote that down somewhere. Yeah, nutrition. Okay, so if you look at these prefixes, so looking at the energy source first, if a bacteria, say an organism, gets its energy from light, okay, so if it gets it from the sun, okay, then we use the prefix photo. Okay, so a phototroph would get its energy from the sun. A chemotroph gets its energy from the breaking of, let me change my color here, from the breaking of bonds. Okay, so usually redox reactions. Okay, so that's a chemotroph. Okay, and then you put those prefixes together with the ones for the carbon source. So if an organism basically gets its carbon just from carbon dioxide, auto, meaning self. Okay, so it prefers itself. And if it gets it from organic compounds, we use the prefix hetero, which means other. Basically, that means other organisms. So something that's a heterotroph means they eat things. So when we put those together, there's really two categories that are the most common. All right, so we'll start with chemoheterotrophs. That's what we are, right? We eat things. We get our energy from the breaking of bonds of the organic things, the other organisms that we eat. Okay, so this is quite popular. We do it, animals do it, lots of bacteria do it, fungi do it, protozoa, very common. The other very common category are the photoautotrophs. So these are anything that photosynthesizes. So they get their energy from the sun, as well as so they're actually able to produce sugars by light energy, and then they fix their carbon dioxide. They get their carbon in the form of carbon dioxide. Okay, so those two are really common. The other two categories, chemoautotrophs and photoheterotrophs, those are mostly bacteria and archaea. I think exclusively bacteria and archaea. They're doing some really kind of bizarre kinds of metabolisms, and I don't want to go too deep in that because it's not really the focus of this course and not something that I want to spend time on. But if you take the upper level microbial physiology, you'll explore some of these kind of weird metabolisms, or else you take a really good environmental microbiology course where your focus is more on these. But the chemoautotrophs, some of these organisms are ones that live in like deep sea vents and just kind of some unusual habitats. They use carbon dioxide to build their organic compounds. And then some of these photoheterotrophs, again, these are bacteria. Some of these bacteria photosynthesize, but they don't produce oxygen. So just kind of some strange stuff there. Okay. So now I want to talk about environmental influences that are going to affect how much bacteria grow. And if your whole lab a week from today is talking about several of these environmental influences and how they're going to change the growth rate of bacteria. Usually the first thing that we think about when we're trying to get bacteria to grow really fast is, do we have the ideal temperature for them to grow? That's kind of the easiest thing sometimes for us to manipulate. But bacteria will have some ideal temperature where they grow the fastest. And usually you get just a little bit higher than that, and that doesn't support growth. Okay, so very quickly begin to die after that ideal temperature. But there's usually a nice range where they will grow. Okay, so for example, I didn't post this. These are 3 different, some kind of solid growth media here. The E. coli has been plated out on, and each of these plates was put at a different temperature. Okay, so the one that was at 37 degrees, which is about body temperature, grew a lot. They incubated one at 30 degrees, a little bit less, and then 22, that's room temperature, grew a little bit, but was really slow. Okay, we left that out at room temperature. It eventually would grow, maybe over a period of 3, 4, 5 days. But in terms of just overnight, you can see that you had the fastest rate at 37. And it's going to be different. A lot of the bacteria that we study in lab, that's kind of the easiest thing for us to do. So just remember that there is a huge variety out there. And of course, we like to slap some kind of definition or term on bacteria describing the temperature that they grow the best at. So most of the organisms that we grow in lab, we consider them mesophiles. Okay, they are in the temperature that they grow at. This is maybe defined a little bit differently, depend on where you look this up. But typically, they're going to grow somewhere between room temperature and body temperature. Okay. So when we're thinking about organisms that cause disease, we are most interested in the mesophiles because they're the ones that grow best at body temperature. There are organisms that grow best at cold temperatures. So we often define cold as maybe being a little bit colder than room temperature, all the way down to freezing. So we refer to those as psychrophiles. And there are organisms that just grow optimally. I mean, they're kind of slow growing, but they grow optimally at polar ice caps or in your refrigerator, for example. And then, of course, there's organisms that like hotter temperatures. We call those thermophiles. These are ones that you're not likely to run into every day because they need temperatures, somewhere above 45 degrees Celsius, which is, above 100 degrees Fahrenheit. So, and then there's all sorts of, we could sort of split hairs. We could say extreme psychrophiles. We could say hypothermophiles. There are bacteria that even grow at boiling, which is it's kind of hard to believe, but there are bacteria that grow really nicely in hot springs, for example. And like I said, we could parse this apart a little bit more. There are organisms that grow best at this temperature, but still grow pretty well at this temperature. So there's other subcategories there that we could talk about. Okay, and you'll test that in lab. You just inoculate some broths and incubate them at different temperatures and see what grows and what doesn't. Pretty straightforward. Oxygen requirement is another big one that we think about. And I showed this media to you before when we were talking about medias and talking about, hey, you might need a special media if you need to get rid of the oxygen so your organism will grow, an anaerobic medium. Well, one thing that you're going to inoculate in lab, if you're in lab, is a thioglycolate test. And basically this is a medium that has thioglycolate in it. No surprise. That's something that reduces any oxygen in the medium to water. Okay, so we call it a reducing medium. Anytime a hydrogen is added to something, it's also carrying electrons as well. So when it gains electrons, it's being reduced. But essentially, we're getting rid of the oxygen in this medium chemically. So with any broth, you expect more oxygen to be at the top than there would be at the bottom. But essentially, we're chemically getting rid of it in that entire broth. So we usually use a oxygen indicator so we can kind of tell the top of the tube where some oxygen has diffused in. So there's always going to be kind of a top layer that has close to atmospheric oxygen in it. And we inoculate our organism and then look to see where it grows. Okay, if it only grows at the top, then we would say, oh, this is an arrow, an obligate arrow. That's an organism that needs oxygen in order for it to reproduce, in order for it to metabolize. Some organisms only would grow at the bottom. We call those obligate anaerobes. Okay? They are poisoned in the presence of oxygen. Now, it's not actually atmospheric oxygen that's a problem. We're going to talk about that in a second. This term, facultative anaerobes, facultative means flexible. Okay, so you can see here's an organism that has grown really well at the top, but it's growing down at the bottom here too. So that's an organism that can use oxygen to respire, but is also capable of using other kinds of either anaerobic respiration or fermentation. So we're going to talk more about metabolism starting next time. There's also organisms considered aerotolerant anaerobes that will grow in the presence of oxygen, will grow without oxygen. They don't really care whether oxygen is there or not because they don't use it. Okay, so with the facultative anaerobe, they're always going to grow faster where there's oxygen, where they're respiring aerobically. Because you just get more energy from that type of metabolism. But an aerotolerant anaerobe, you can see it's growing the same whether it's in the presence of oxygen or not. So in practice, it's kind of difficult to tell the difference between the last two tubes there, but at least those categories exist. Okay. So I said it's not really atmospheric oxygen that's a problem. I want to talk about that a little bit. I want to talk about toxic forms of oxygen because we're going to be revisiting this a few times throughout the semester. So I'm not super interested in the chemistry here, so don't worry about the notation. But I want you to think about three things. These toxic forms of oxygen, why are they bad? Where do they come from? And what can cells do to counteract it? So what can cells do to, we'll say, to overcome being in the presence of some toxic oxygen? Okay. So first of all, why are these forms of oxygen bad? They're all forms of oxygen that have their electrons distributed inappropriately, or else they have an unpaired electron, like the hydroxyl radical. That's the worst one because it has an unpaired electron. When these electrons are not distributed appropriately, they are trying to grab electrons from other molecules. Okay, so we say that they are all strong oxidizing agents. And that means, like I said, they are stealing electrons from other molecules. Ran out of room there. So in other words, they're causing all kinds of damage to structures and cells. DNA, proteins, all kinds, lots of damage because they're stealing the electrons. Well, where do they come from? This singlet oxygen, this is most often formed during photosynthesis, and it's a result of oxygen being raised up to a higher energy state. So that's not typically something, you know, certainly the bacteria that are pathogens would encounter very often. Plants would have to deal with this, and they would produce pigments like carotenoids to counteract. Okay, so we'll say pigments would counteract. Okay. And then let me skip to the bottom one here, hydroxyl radicals. Where would cells encounter that? That's produced as a result of ionizing radiation. And we're going to talk a lot more about non-ionizing radiation and ionizing radiation. Basically, ionizing radiation is being exposed to X-rays or gamma rays. Okay, so that's not going to happen really all that often. So wouldn't likely to encounter that, so they don't really have a particular mechanism to counteract that. But the two kinds of toxic form of oxygen that they are likely to encounter is superoxide and peroxide anion. Because both of those are formed during aerobic respiration. Okay, and when we talk in more detail about respiration, again, starting on Wednesday, I'll point out when that likely happens during electron transport chains is when that happens. So basically, any cell that's going to be in the air is going to be exposed to superoxide or peroxide anion, just as because they're around other organisms that are respiring aerobically. Okay, so superoxide. I don't know why I'm getting that. That's what the problem was. Sorry, this recording may not be that great. So what do cells do about superoxide and peroxide anion? Our cells have to deal with these toxic forms of oxygen as well. And I don't have this formula memorized, so. But the solution for cells is to produce an enzyme to counteract it. So superoxide dismutase will detoxify this superoxide into hydrogen peroxide. Now, the problem with that is hydrogen peroxide has peroxide anion in it, so that's still toxic. So there needs to be other enzymes that are going to detoxify hydrogen peroxide. One of them is called catalase, and that detoxifies hydrogen peroxide to water and oxygen. Balance equation. Okay. And I'll say or peroxidase. That's another enzyme that detoxifies hydrogen peroxide. You get a little different end products. I wanted to make sure I mentioned catalase because this is a test, again, that if you are in lab, you will do later on in the semester. We rub some cells onto a glass slide and then add a drop of hydrogen peroxide. So if it bubbles, if we get the release of the product there, then we know that the organism must be detoxifying that hydrogen peroxide by producing catalase. So it's a nice, easy way to tell the difference between an organism that makes catalase and an organism that doesn't. There's some closely related organism that's really helpful for. So in a nutshell, if an organism can make superoxide dismutase and either catalase or peroxidase, then it's more likely to be able to grow in the presence of oxygen. So it's more likely to be a facultative anaerobe or an obligate anaerobe. Okay, and even a cell that was an aerotolerant anaerobe, even though it didn't use oxygen, it would need to be able to live in the presence of it, so it would have to have some of those detoxifying enzymes. Okay, an organism like Clostridium tetani that is an obligate anaerobe would not be able to make it's obligate anaerobe because it can't make those detoxifying enzymes. Okay. All right, moving on. Stop me if there's questions. Okay. pH. Okay, obviously organisms are going to have a kind of ideal hydrogen ion concentration that they're going to, that their enzymes are going to work best at. This is a little more straightforward. An acidophile will grow best at acid pH. A neutrophile at neutral pH. An alkalinophile or alkalophile at alkaline pH. So Some examples, there are lots of acidophiles that would live, Yellowstone hot springs, where you've got kind of, funky mineral, different kinds of organisms growing in that hot water. Acid runoff from old mines, for example, or in your stomach. pHs can be as low as 2 in your stomach, and there are bacteria that live there quite comfortably. Most of the organisms that we work with are going to be nutrophiles. The best example of an alkalinophile that I could think of is Vibrio cholerae. The causative agent of cholera actually grows best at pH of about 9. And there are, various bodies of water throughout the world that actually have a little bit more alkaline pH. Okay. All right, and physical effects of water. So we talked about osmosis and reviewed that a couple of lectures ago. Organisms, they're may not grow well if they are in a environment that has a lot of solutes, especially. So we call that pressure osmotic pressure. Now, don't confuse this with barometric pressure, not atmospheric pressure. So there are barophiles. These are organisms that grow best like at the bottom of the ocean. We're talking about here, when we talk about osmotic pressure, the solute concentration in the solution that a cell is in. And when you look this up to define it, you get a lot of definitions that are rather difficult to kind of understand. An easier way to think about this is how badly water wants to leave the cell. That's one way that I've seen it defined. Now, it is possible. So if you've got your cell in the solution that's hypertonic, right? So lots of solutes, okay, then water wants to leave that cell really, really bad because it wants to equilibrate the solute concentration inside and outside the cell. That's usually the really bad environment for bacteria because they're going to dehydrate. Potentially, right, it would be really bad if you had a huge gradient the other way, but that just doesn't happen as commonly in nature. And plus, remember, our bacteria cells have a nice thick cell wall that's going to keep them from bursting. Now, if the cell wall is not intact, then that would be bad, even maybe a slight gradient, but an intact cell wall. Usually when we're talking about high osmotic pressure, we're talking about increasingly more solutes in the outside of the cell. So increasingly hypertonic. All right, so you're going to test this in lab. You're going to test and see if your organism grows in 5% salt. Will it grow in 10% salt solution, 25% salt solution? Now, there are organisms, most organisms can't survive that, again, because they're losing water. When we try to keep bacteria from growing on our food, for example, we might add, preserved meat and salt. Not because we're trying to preserve the meat, we're trying to keep the bacteria from growing on the meat. All right, so high salt solution, most bacteria can't survive, although there's exceptions. Osmophiles are organisms that grow best in a high solute concentration. There are organisms that have, mechanisms to do that or to pump water one way or the other. And then a halophile is a osmophile that grows best specifically when the solute is salt. So we'll say high salt concentration. When we put brackets around something, that means concentration. Okay, so there are organisms, say, that live in the Great Salt Lake, where salinity can reach like 25%. There are organisms that can only live there and wouldn't be able to live in a more isotonic solution. So, again, bacteria basically are able to live just about anywhere. Okay, and one other environmental thing that I wanted to bring up that they don't talk about in the book are ecological associations. So sometimes when we're trying to get bacteria to grow in the lab, we are not successful because they need to be in some kind of relationship with another organism in order to support their growth. Okay, one of the main reasons I talk about this is because this is a really hot topic in microbiological research, especially before the pandemic, human microbiome, right? So the study of the interactions of all the bacteria in our body and how we can manipulate that to make us more healthy. Like we know that when certain kinds of bacteria are in our guts, that can make people more likely to be hungry, more likely to be obese, more likely to have inflammation in their gut. So if we understood the ecological interactions of those bacteria, then hopefully we would be healthy. We don't right now. So probiotics, you can take probiotics and we have no idea whether they'll work or not. But hopefully someday we can dial it in and say, hey, I'm going to swallow this bacteria, and then that's going to help, this part of me be more healthy. All right, so we know that organisms are always living in association with other species. We could describe those relationships as antagonistic, meaning one organism benefits and one is hindered. Okay, so we could also refer to that as a parasitic relationship. And here's an example of an antagonistic relationship between this mold, this white mold right here, and this is some bacteria that's streaked on a plate. You can see there's a little, well, I put a scribble down there now, but there's a little zone there around the mold where the bacteria is not growing. That's because the mold has secreted something that inhibits the growth of bacteria. So in this case, I'm not sure that the mold is benefited from the bacteria, but the bacteria is definitely inhibited by the mold. It's secreting some kind of antibiotic there. A synergetic relationship is a little bit harder to think of an example of that, certainly from the bacterial world. But this is when one organism, well, let's say two organisms benefit, or both organisms benefit from their relationship with each other. But they are still able to live separately. So members could live separately, but they usually benefit from the association with each other. So we know that bacteria sometimes live in biofilms, in big mats of communities of organisms, sometimes not. Sometimes they might benefit from that. Again, that's a little bit harder to parse out and give an example. I have lots of examples of symbiotic relationships. When two organisms, they benefit so much that they rarely live separately because they can't survive without each other. Okay. In other words, okay, so both organisms benefit, one rarely can live without the other. So we have bacteria that live in the guts of termites. Okay, those bacteria are what digests the wood that the termites eat. Okay, so the termite gives the bacteria a nice warm place, lots of nutrients, and in turn, the bacteria makes those nutrients available to the termite. Bacteria in the rumen of cows, right? Cows can digest grass without those bacteria. This picture here, these nodules on the roots of this pea plant there's actually bacteria in there and those nodules that are fixing nitrogen into a form so that the pea plant, it's available to the pea plant. Okay, so the bacteria gives the nitrogen that it needs to the pea plant, and in turn, the pea plant provides nutrients for energy for the bacterium. Okay. All right, so let's talk some detail about the actual growth of microbes. So how can we describe what's actually happening there? Typically, when we talk about growth, we are to some extent talking about bacteria cells getting bigger, but typically what we're referring to is that they are growing in number. Okay, so when a bacteria cell grows and divides, we call that binary fission. So again, one cell gets larger and divides in two. Right, so sometimes when you're looking at a bacterial culture, you'll see rods, for example. You see some short ones and you see some long ones. That still can be the same species, but some of them are just getting ready to divide. So the longer ones are getting ready to divide. They'll elongate. You'll see a septum form there, or you see that here in this transmission electron micrograph. Those sides pinch off, and then that parent cell becomes two daughter cells. That's binary fission, and that's pretty much the way bacteria divide. The time it takes for them to do that, we call generation time. Right, so time it takes for one bacteria to become two, also known as doubling time. And there's a lot of variability between species, how long it takes for one bacteria to become two. That was one lab that I can remember quite vividly during my undergraduate days. We had taken a slide, a bacterial slide, and then we had actually put a agar medium, kind of painted it on there, onto the slide, and then inoculated our bacteria onto that growth medium. And then under the microscope, our job, we were supposed to sit there and focus on a single cell and just watch it and wait to see and watch it grow. So we were supposed to sit there for, however long it took for that cell to divide and become two cells. And I remember thinking that was like the dumbest thing ever, and it was so boring, but it's like the only thing I remember from that class now. So now I think it's a little bit more interesting. But this shows you then graphically the doubling time. So here's one cell, and the time it takes for one to become two. This is the generation time. So in this example, it's 30 minutes. Under ideal conditions, E. coli will take somewhere between 20 and 25 minutes for one cell to become two. But a lot of variability there. Here's another generation. We've got two cells and they become four. Okay, 30 plus 30, 60 minutes. Four cells, pretend this is 8. I don't know why they drew 9 there, but okay, but cells double with each generation. So 4 cells become 8, 8 cells become 16. So you get the idea. That's what we call exponential growth. When a population will double every generation. So we can represent that graphically. Exponential growth, or another name for that is logarithmic growth. Okay, it's gonna, when you graph out, every generation and the numbers here on the y-axis, then you'll get a curve that really quickly rises up really quickly. If we were describing arithmetic growth, that describes a population that will only gain a constant number like every generation. So if they're adding, you know, one new organism every generation, that's going to increase very, very slowly. Okay, generally populations are going to, again, behave exponential unless there's something that's really limiting that. So if we think about the generation time, that's dependent on, again, it depends on what species. Like the mycobacterium that we stained in lab this morning, they typically have a really slow generation time. It takes them a long time to make that mycolic acid in their cell walls. So it does vary just depending on the species. It also is going to depend on environmental factors like nutrient availability. Temperature, pH, all those other things that we just talked about to some extent, whether those conditions are just right or not. Okay. So again, thinking about representing growth, you know, it's very difficult to represent exponential growth on a regular graph paper, because if you tried to plot this, right, it's doubling every whatever, however many, 20 minutes, 30 minutes, whatever. You really quickly get into really big numbers. So typically, if we were going to express this graphically, we would use semi-log paper. So semi-log paper means that one axis has a logarithmic scale. So that means it's going to have 10 to the 2, that's the same thing as 100, 1,000, it's like adding a 0 every unit there. So this way, we could plot out our counts in kind of on one page. And this way is nice too. If you know, say you take counts over an hour or two, and then you want to know, well, how many bacteria am I going to have at, you know, say 6 or 7 hours? You could extrapolate back just from the graph to figure out, oh, about how many numbers I would have that way. Okay, so that would be very difficult to do. What do I, you know, what's this going to be up here if you had kind of regular graph paper? All right, so we have a little time left. I want to talk about how we can calculate cell numbers just kind of by knowing a few things, by knowing how many we have to begin with, and maybe the generation time or how many generations we have to go. So I want to kind of show you how to do this, like with the mathematical formula first, and then another way to kind of think through it, which is pretty easy to do if the numbers are not too crazy. So first thing to understand that when we're trying to understand the number of cells arising from a single cell, we express that as 2 to the n, where n means the number of generations. Because it's 2, because it's doubling every generation. All right, so if we have, again, we have we start with a single cell, and if we have, you know, 1 generation, right, that's going to equal 2 cells. Again, starting from a single cell in 2 generations, 2 times 2, that's going to equal 4. Okay, 3 generations, 2 times 2 times 2, that's going to equal 8, right? So the formula that the book gives you is pretty straightforward. The final number, F means final, of cells is going, sorry, I don't want to say that backwards. Yeah, is equal to the initial number of cells times 2 to the n. So basically n, remember, is the number of generations. All right, so if we wanted to figure out the final number of cells, and say we, say you begin a culture with 100 cells, okay? How many in 3 generations? Okay, because we have low numbers, we can do this in our head. Okay, so we started with 100 cells, multiply by 2 to the third. Okay, so 2 times 2 times 2 is 8 times 100. Okay, that is going to equal 800 cells. That's our final number of cells. Okay, so now we really don't need a fancy formula to do this. If we're going to start with 100 cells, that's our time 0. Over 1 generation, we're going to have 200. We just double it. 2 generations, 400. 3 generations, 800. Okay, so you can kind of think through it and doubling it that way. Okay, so I want to give you a practice question. If you have, we've run out of time. So if you have time, take a look at this. We will go through it in class next time. There are a couple homework questions that are almost exactly like this. So doing what we did, if you have a chance, take a look at that, and then we'll do it together next class. If you want to look at your exam, I'm available for a while, or come see me during office hours tomorrow.